Hello everyone, I'm very happy to present our research we conducted at a global scale, titled Warmer and Drier Ecosystems Select for Smaller Bacterial Genomes in Global Soils. Background introduction. Trait-based approaches have revolutionized ecology and provide demonstrable value to our ecological understanding of plants and animals. However, we know little about the biogeographic pattern of microbial traits across the globe. Among those well-defined microbial traits, genome size is fundamental for the prediction of microbial physiology, their ecological relationships with other microbes and the environment, and their evolutionary histories. This is because genome size is linked to other bacterial traits related to life history strategies. For example, growth rates and tolerance to extreme conditions. Variation in genome size among microbial taxa can reflect evolutionary events such as genome expansion and reduction that may be associated with emerging of new species, functions, or lifestyles, and organisms with large genomes require more resources to grow and reproduce and can have a more restricted ecological distribution. Soil functions are strictly dependent on critical factors like water, light, and biodiversity. Recent studies have investigated microbial genome size in response to ecosystem type, trophic strategy, and oxygen use, but investigations in soil microbial genomes and their responses to environmental factors along large spatial gradients are still limited. In theory, both a trade-off and a unidirectional pattern can exist in scale-up or global-scale environmental investigations. Such environmental effects on microbial species occurrences can be either positive, negative, or neutral, and the magnitude of the effect differs among bacterial species. In this study, we propose 4 types of relationships between environmental effects and bacterial genome size. We proposed trade-off, unidirectional effects, and no correlations. In this study, we aim to investigate whether genome size could mediate effects of environmental gradients on bacterial occurrences at the global scale. To test the above hypothesis, we analyzed bacterial communities of soil samples collected from 237 locations across the globe, covering different ecosystem types and diverse environmental gradients, such as soil aridity, nutrients, and air temperature. In total, 2,315 genera were detected using 16S RNA gene amplicon sequencing, and bacterial features annotated at the genus level were extracted. We then matched our global soil microbiome data with bacterial genera in the publicly available database, resulting in 143 bacterial genera with genome size values ranging from 1.58 megabase pairs to 0.4 megabase pairs. We then investigated relationships between the environmental gradients and bacterial genome size by fitting the dataset with a joint species distribution model. After fitting the model, we found that the distance from the equator had a strong impact on genome size distribution in soil, in that bacteria with small genomes were more likely to have a positive association with increasing latitude. Importantly, we found that bacterial genome size significantly correlated with aridity, in that bacteria with small genomes exhibited positive responses to increasing aridity, while bacterial genera with large genomes tend not to exhibit any responses. This trend of bacterial selection with small genomes was consistent across different taxonomy. In summary, our study provides novel evidence that environmental stress associated with increases in aridity and warmer climates favors bacteria with smaller genomes in soil, despite the unknown consequences of such microbial changes to ecosystem functions. Future climate change research and policy making should consider soil bacteria and their functional traits, as they play important roles in soil carbon sequestration and maintaining ecosystem functions. Future research is also warranted to validate our findings by investigating bacterial community assembly in soil and other ecosystems by determining genome size from metagenomes across environmental gradients.